and share my screen. Okay, so I think this is about where we stopped last time, right? Just trying to increase the size of this screen that have you guys on, so I can see more of you. Hello, Anish. Ah, okay. We were talking about homologous structures, and we stop at this slide where we say that in this case, the forelimb of mouse and bats are start equally. So we see here the limb zone buds. And then as time passes by, we see that the membrane for the bats remain, but for the mouse is lost. So in this case, this is a great example of homologous and anatomical structures that can be differentiated by altering the development, whether the membrane is lost or is not. Okay, where is here? Okay. So now, in the medical, in embryology and overall, developmental biology has become a really important branch in medicine because we can, physicians became interested in embryology from a practical standpoint. What do you think is the percentage of births, human infants, that are born with an anatomical abnormality? Just throw a number.
10%.
10%. Okay. Oh my goodness, that's a little high. But yeah, so normally it's a little high, but not far. So between 2 to 5% of infants are born with a defect, which, if you look at it, is devastating for the couple, right? For the parents that get this. So those abnormalities can vary of having no arms, fingers, heart problems, eye problems, skin problems, you name it. There is so many diseases and embryological defects nowadays, and more now. So information on the causes of the birth defects help doctors to counsel parents, predict the chances of additional children having defects. Because if you have one, you need to investigate why that baby was born with a defect, just to prevent, maybe, if possible, that the second kid gets any issues, right? For those that are interested into going into genetic counseling, this is one of the courses that you may require and is important to have, okay? So we need also to understand human development. And in order to do that, we have a little bit of problems sometimes, because ethics, right? It's easier to experiment with a mouse like that with a baby, right? Or an unborn baby. So ethics prevent us from using humans as model organisms, and hence we need to turn our face into models that can have some genetic background that is very similar to humans, or that the toolbox that I've been talking about since Monday works in a similar manner. So when we talk about medical embryology and teratology, there are two aspects that are important. One is malformation, and the other one is syndromes. So malformations are abnormalities caused by genetic events. These genetic events could be gene mutations, aneuploidies, abnormal number of chromosomes, or chromosomal translocations too, okay? So there's many things that can cause, but are mainly genetic events. Syndromes are the expression of malformations. So malformation often appear as syndromes. What that it means? That we can have several abnormalities that can occur at once. Okay, like the image that we have in here is called Piebaldism. Okay, Piebaldism is caused by a dominant mutation on the KIT gene. The syndrome includes anemia, includes sterility, includes these patches where the pigmentation does not occur in the middle line of the abdomen and in the front head part of it. Okay, it also includes deafness and the absence of nerves in the gut. So how many I named? 4, 5, 6 different malformations, and that is a syndrome. Okay, so you're going to have many. Oh, okay, the alarm is going on. Okay. That occurs at the same time. Okay, so, and these features appear in 2 different organisms, a mouse and a baby human. Okay, so this is where some of these conditions can be studied using other model organisms. Okay, so organs that are linked in developmental syndromes share a common origin of mechanism of formation, and that's the type of things that we're going to be looking through this course. Okay. Okay. Now, disruptions. Now, this is the part of the teratology. Okay. Teratology is the study of how environmental agents disrupt normal development. And nowadays, we can't escape of being subjected to a pollutant, to a molecule that can dramatically affect development. Okay? So this disruption is going to cause different abnormalities that are caused by exogenous molecules called teratogens. Some can be chemical, some can be viruses, some can be exposure to radiation, hyperthermia, for instance. Okay. The example that we have in this image is called phocomelia, first of all. So it's a developmental abnormality caused by thalidomide. Thalidomide was taken by mothers during pregnancy to supposedly avoid the morning sickness. It took years before that was taken down or out of the market. And Canada was one of the worst. It took years for it. Okay, so thalidomide is a potent teratogen. It has been tested in zebrafish, in chickens, in rabbits, in primates, and also, of course, in humans. Okay, so it causes, we can see from this photo, severe birth defects. Okay, some can have the absence of ears. It all depends on what time the mother started taking the thalidomide. Okay, we can have, and this is days after last menstruation. It's not days of pregnancy, it's last menstruation. Okay, the absence of thumbs, absence of arms, severe shortness of arms. That is what this individual has. Okay, dislocation of the hip. That is also what this individual has. Malformation of ear. There is another photo that I have. I took it out this year, but they're missing the ears, or the ears are weird formed. Okay, the absence of legs, severe shortness of legs. That is also what this individual have, and malformed thumbs. So it can occur. It all depends on what time, which means that there is a window in which this substance is capable of causing all these transformations. This thalidomide was sold in a number of countries across the world from 1957 to 1961. So it was just a couple of years. And in 61 was withdrawn from the market after being found that it caused birth defects. And we now know that it has even the third and fourth generation of people that use the thalidomide can have defects. So it's transgenerational. It's not just he took it, he got his mother took it, he got the problems, and now he can have babies with no trouble. His kids are most likely to also suffer other conditions just because of this. This use of thalidomide is one of the biggest tragedies in modern times. We believe that there is an estimate between 10 to 20,000 kids that got affected by this drug. So now we have a little bit more of control of drugs. We have the FDA in the states that approve whether the drugs are safe or not. We still use this drug, but for treating cancer, not necessarily for avoiding morning sickness. So we want to watch a little video that resumes a little bit what happened with this drug.
Dana-Farber Cancer Institute chemical biology researchers solve a 60-year-old medical mystery that will now help guide new drug development. More than 10,000 children in the 1950s were born with severe birth defects, and there were even more miscarriages after their mothers were prescribed a drug named thalidomide to treat morning sickness. Today, thalidomide is not given to pregnant women, but it is used as a powerful cancer drug, primarily in the treatment of multiple myeloma to inhibit tumor growth. Scientists never knew why it caused birth defects until now. Senior study author Dr. Eric Fisher of Dana-Farber says the research team linked the defects to the disruption of a key protein involved in fetal development called SOL4.
What it really means is that we have the puzzle pieces in place, so we for the first time understand what all the targets of these drugs are.
And that means drug developers can use this finding to guide new drug development where the benefits are great without the harm for devastating birth defects. The study published online by the journal eLife.
So sometimes we take drugs, we take things, and we don't know necessarily what the effects are. And later on, we learn that it was devastating. So anyways, and that is the end of this chapter. Where is my mouse? I have a hard time going from one screen to the other. And we're gonna switch to the next chapter. Just need to open that one. Come on. I am using a different mouse for my left hand, and I am not as good at it to manipulate, so bear with me. Okay. So in this chapter, is we're going to be talking a little bit about developmental patterns. So it's chapter 2. So early studies in embryology had their roots in anatomy. So it was describing, looking at evolutionary embryology, things like that. And most of the questions were about what this becomes, what this part do. Okay. But at the end of the 19th century, as we saw on Monday, embryologists start asking the question, how? Okay. How this developmental process occur? How this part is created? Okay. How does an egg become an adult? Thus, the field of physiological embryology was born because we talk about the how, the physiology, how this works, how this functions. So the physiological embryology goal is to find the molecules and processes that cause the visible changes in embryo, like we see in here. So, okay, this is the brain start developing, it's like huge, and then it start changing. Okay, so in order to do that, well, they needed to do a lot of experimentation. So experimentation was added to the observation in order to determine how embryonic cells respond to perturbation and disruptions. So to do that, they initially started using 4 different techniques, and some of the techniques that we have in front of the screen. So we have a defect experiment. These defect experiments, for that, what they did is that they used a portion of the embryo that was destroyed, and then they observe the development of that embryo and see what happens. Is the embryo well at the end? Yes or no? Okay. They also use isolation experiments. These isolation experiments is that the portion of the embryo is removed, and then the development of the partial embryo and the isolated part is observed. Okay, so let's see. If I take the cell, what the cell will become, and if the embryo is going to be normal or is going to miss the section that we took out. They also do recombination experiments. This recombination experiment is looking at the development of the embryo and observation after replacing an original part with a part from a different region of the embryo. So like creating Frankensteins, right? I'm going to take this part, I'm going to put it here, and let's see what happens. And then is the transplantation. The transplantation is a portion of an embryo is replaced with the portion of a different embryo. So that's the main difference between recombination and transplantation. Recombination is using parts of the same embryo, and transplantation is using parts of a different embryo. So as cells start forming, because we know that we got one day that is the fusion from the sperm and the egg, and then we have a zygote, and then they start multiplying, right? So as cells start forming, there's a lot of things that can occur, and it's just amazing. Okay, so here we have in this table the type of cell, differential cell product, and specialized function. So a keratinocyte is an epidermal cell. It can form keratin, and their function is the protection against abrasion. We have the lens cells, for instance. They are formed the crystallines, and their function is the transmission of light. The melanocyte is the melanin and is a pigment product. So we have a whole lot of cells, right, that can accomplish specific functions, and they're going to have different origins too sometimes. So cells need to become organized in order to become tissues, and tissues then gonna form organs, and then organs can form systems until finally we have an embryo. But how these cells know where to go and how to communicate to become an eye, for instance. There should be signals, right? That tells them, okay, you are placed at this time here, you are to become this. So they don't decide, right? Or do they? Do you think that cells decide to become a cell from your nose or your heart or your eye? Decision. We always associate decision with a brain. No? The brain takes a decision. We consciously take a decision, but a cell doesn't have a brain, but it's going to form something, right? So, well, these decisions, and I put in these decisions in quotation marks, okay, made by the cell is what we call choose a fate, okay? And is determined by the level of commitment of the cell. So a cell starts with a generalized group of cells, and then depending on the factors, molecules surrounding, who is on my side, do I like you, do I don't like you, we commit, okay? It's like you guys may be in that age that you have a boyfriend, girlfriend, okay? And you have spent a certain amount of time together, and then one of you decide that this is it. And then you throw the sentence, right? Will you be with me for the rest of my life? Utopic, but let's see. We all hope for a long life together, right? And then you commit to that person. Well, it's the same case here for the cells. The commitment, the level of commitment of a cell, depending on the surrounding, depending on the condition, is the same as a couple, right? Depending on the condition, depending whether that, how many things you have, the affinity for the other person, you decide this is it. Well, the cells do the same. They commit. So depending on the level of commitment, they decide to become one of these type of cells that our body has produced. So cell specification has been tested through experimentation in embryology and had been able to learn how interactions between embryonic cells generates the embryo, how these interactions occur, okay? One of the first concepts that I want you to retain is that development of specialized cells is called differentiation, okay? Development of specialized cells is what we call differentiation. Once the cells stop their division process, they will develop specialized structure elements and also will accomplish different functional properties. So by undergoing different changes, the cells start the process of becoming a specific cell type. These changes in cellular biochemistry and function are preceded by a process called commitment. And that's what I talked a couple of minutes ago, to the cell, to a certain fate. So cells at this point may not look any different. However, their developmental fate has become restricted. So the process of commitment of a cell can be divided into two different stages. Okay, a specification. This specification stage is labile. Okay, so at this stage, the commitment is still capable of being reversed. Okay, so this is how somebody has asked you to be their partner for the rest of their life, and then life goes on, and then that person become a little different of what you were before, and then you decided to break that commitment, right? So it's reversible. Even though you were specified to be together, you can change your mind and say, I don't think so, okay? The second stage is the determination. The commitment is irreversible at this point. Even if a cell is placed in another region of the embryo, it still becomes what it was specified to become, regardless of the new environment in which it finds itself. So let's see this in figures. So the fate of a cell or tissue is said to be specified when it's capable of differentiating autonomously, okay? Meaning by itself, when placed in an environment that is neutral with respect to the development pathway, like in a petri dish, as we have in here. So we have here a mass of cells. Some are yellow, some are pink reddish. We take one yellow, put it in here. We put one red, put it in here. These were specified to become muscle, and then they become muscle. These were specified to become a neuron, and they became a neuron. So once again, at this stage of commitment, cells are labile, are capable of being altered, because here we haven't specified it yet, okay? We take them here, and then we give some specifications, and then they become. Now, if a specific cell is transplanted to a population of different specified cells, the fate of the transplant will be altered by its interaction with their neighbors, okay? And this is what we see in this figure, okay? So here in B, we take these cells, we have majority cells that are yellow, and only one that is to become supposedly a muscle, okay? So in this scenario, the red cells, even though they were specified, right, but not determined, just be careful with the two terms, specified and determined, to form a muscle, as in the previous image. However, under the new circumstances, because it has been surrounded by nervous cells, okay, this blastula is influenced by those neighbors, okay? So even though it was specified to become a muscle, it became a neuron, because the surrounding of the other cells are telling you, no, you gotta become this, okay? In C, what we have in here is the second stage of commitment, okay? So here they are committed already. They are determined already to become. So in this figure, we see that if the red cell was committed and determined to become muscle in the time of the cluttering, okay, then it will continue to differentiate into a muscle fiber, that's what it became, despite of the interactions that they had with the neighbors. So at this point, it is irreversible. So determination, which the second stage of commitment, okay? So it all depends at what time these cells are placed together. Hence, during embryogenesis, cells need to mature through specific stages that cumulatively commit to a specific fate. So we have those three steps: a specification, determination, and differentiation. During a specification, there are three further major strategies. The strategy used depend on the animal species, okay? So we have autonomous specification, we have conditional specification, and we have syncytial specification. It all depends, but we're gonna go through each of them and one at a time. So let's start with the autonomous specification. So in this autonomous specification, we see that this is characteristic of most invertebrates. Okay, we don't see this much into vertebrates, but we see it more in invertebrates. So in an embryo, depending on the cell position in the egg, cells have a specific fate. So here is the normal development of patella. Okay, and we have cells blue and cells that are pink. Okay, so the colors are indicating that they are supposed to become something. Okay, and their position is very strategic in this embryo because it all depends in the pattern or segmentation. Okay, so these morphogenic determinants will influence the fate, the cell fate, and are often called transcription factors. Okay, and these are found in the egg cytoplasm. We're going to be talking about that later, okay, as we advance in the course. So transcription factors are the ones that are going to kind of influence that cell fate. But let's don't worry about this for now. So let's look at the figure more in detail. So if a particular blastomere, remember that a blastomere is one of the cells of the early embryo, okay, one start dividing. So if we take one blastomere, here we took this one, we put it here, and we produce the same type of cell that it would have made if it was still part of the embryo. In addition, the embryo from which the blastomere was taken will lack those cells, and only those cells that will have been produced if the blastomere wasn't missing. Okay, so in A, in here, is a 16-cell stage cell. Okay, and cells are normally to become the one that is pink, to become a ciliated cell that is called the trochoblast. Okay, this is the presumptive trochoblast. In B, we have a 48-stage cell, and in C, we have a ciliated larval cell. So we have all the cells in here. Okay, and D through G, we have differentiated the Patella trochoblast. So we took that trochoblast and we isolated. And because they were predetermined to become the trochoblast, that's what they became. So they became the correct type of cell because it has no influence whatsoever from anybody else or any other cell around. So this is autonomous specification. So recent studies show that tunicate larvae have autonomous specification. That's what we see in the image. As we can see on the left-hand figure, the embryo of the tunicate rises from cells with the yellow pigment. So we have here our egg, we have the crescent, the yellow crescent, and then we start having some segmentation, right? Depending on the position that these cells are taking, whether they are more toward the animal pole or the vegetal pole, they are like predetermined to become something else. Okay. In this figure B, what we see is that even at the stage one cell, this is one cell, okay, and this is one cell, animal pole and vegetal pole, okay. The ones that are here are predetermined to become more ectoderm, closest to the midline. After some segmentation, we're going to have the nervous system. And in the vegetal pole, what we have is more endoderm and mesoderm. So just based on the position of those blastomeres in the early embryo, they are already predetermined to a certain point to become something. And this is here only a stage of 8 cells. So 1, 2, 3, 4, 6, 5, and 8. Okay, 8 cells embryo. We have already a predetermined regions of what these cells will become. Okay, in this figure C over here, we see that in the tunicate with the color indicating tissues, so we have the neural tube is in yellow, is all the way down, okay, and green is the notochord, very important during development, as we're going to see. Then in blue here, we have the endoderm, and epidermis, of course, all around. So each cell is predetermined already to become something. Now, the fate map shows that when the 4 blastomere pairs of the 8 cells are dissociated, so they took all those cells. So these are this group here, these ones are this group here, and so on. We dissociate them. We don't allow them to communicate. It will form the tissues that they were supposed to form if they remain in the embryo intact. Okay, so this type of experiments reinforce the idea of the existence of cytoplasmic segregation of tissues that are determined way before they can even communicate to one another. And those factors are in the egg. And depending on the position, then this is where that even if we segregate the cells, they become what they were supposed to. Now, they may not be completely functional, okay, because they need to communicate to the other parts to form a real embryo. Okay, so the second type of a specification is the conditional specification. This conditional specification is present in some invertebrates and especially in vertebrates. Okay, so conditional specification is a process in which cells achieve their respective fates by interacting with other cells. So they require other cells to tell them what to become. So it is called conditional specification because the fate of a cell depends on the conditions in which the cells find itself. So there are different ways how we can, how they can interact. Either cell-to-cell contact. When we have cell-to-cell direct interaction, they depend on Juxtacrine factors. So start keeping that Juxtacrine name in there. Tomorrow we want to be talking more about these things. So cell-to-cell direct contact depends on Juxtacrine factors. Okay. Some will also depend on secreted signals, and we call these ones paracrine factors. And some will depend on physical properties of the environment. So in this image, what we see is a blastomere, okay, that has been removed from, and this is key, an early embryo, okay? So we are in a blastula. Blastula is an early embryo. So we took a group of cells here, okay? And what we did is that the remaining embryonic cells altered their fates so that the roles of the missing cells can be taken over. So we took them and we produced an embryo that was no missing anything at all, okay? Because the cells that surrounded the section that was taken up took over. And they say like, oh, geez, something is missing. Okay, so because we are not completely determined yet, we can replace that and then we can form a full embryo. Okay, so This ability of the embryonic cells to change their fate help to compensate for the missing part, and that is called regulation. Okay, so the cells that remain control to replace and regulate the creation of the embryo. So in here, we took that and we transplant and put it in a completely different part, right? This is the animal pole, this is the vegetal pole, and the embryo, these cells simply became what they were supposed based on the environment that they were now, right? So they became cells of the belly tissue. So in either case, these ones didn't miss anything, and when they replaced into the other embryo, because it's a transplantation, then this embryo didn't have any part that was supposed to be forming if they were remaining here, but it became a belly section. So when a cell becomes dependent on the position of the embryo, and its fate is determined by the interaction with the neighboring cells. That's what we see in this A section, okay? In B, what we have in here is if the cells are removed from the embryo, the remaining cells can regulate and compensate the missing part. So we took it, we didn't replace it, and we have a full embryo. Nothing happened, okay? Thus, because of this mode of specification, an isolated blastomers can give rise to a wide variety of cells. A conditional specification is said to give rise to pattern of embryogenesis called regulative development. And it's seen in most vertebrate embryos. So if early development, we can take some parts, nothing happens. And this is something that is exploited for in vitro fertilization, for instance, okay? Because we have an embryo that has been frozen, but most of the parents that go through in vitro fertilization would like to know if the embryo has any diseases, right? You don't want necessarily to have a baby that will have a condition. So they're going to take one of the blastomeres of that zygote and that embryo that has been frozen to check if they have any abnormalities, any gene that can produce a baby with a malformation. And the babies are perfectly fine, even though they took 1 or 2 blastomeres from the in vitro embryo, okay? Okay, the last type of specification is the syncytial specification. It's characteristic of most insect classes, and this type of specification uses both autonomous and conditional elements. So in here we have that a cytoplasm contains many nuclei that is called the syncytium, and this is what we see here. All of this is the syncytium. So we have many nuclei inside. Hence, the specification is presumptive cells within the syncytium is called syncytial specification because it has the syncytium that is in there, okay? So cells are committed to the fate based on interactions among the parts of that cell. So in early embryos, cell division is not complete, and they are always communicating with the cytoplasm. So cells, part of the cytoplasm of the cell is connected to the syncytium here, okay? So the nuclei divide within the egg of the cytoplasm, creating many nuclei as we see time pass by. But also, with time, the cytoplasm of the egg is not uniform. And the anterior and posterior parts have different molecules that will indicate which part these cells are going to form compared to these parts. So we see here different colors, okay, that allows to see a gradient. So in this case, we depend of a gradient. And this is what we see here in this image. And this image is the example using the Drosophila melanogaster, the fruit fly. And we depend on morphogenes, okay? So each time that the nuclei divide, the position of the nuclei is carefully maintained across the syncytial blastoderm. So here we have a bicoid cytoplasma, greater concentration in the anterior part, lesser concentration in the posterior part. And then here we have the gradient for the caudal part that is high at the back and very little in the front. So as cells start dividing and the nuclei start maintaining a position within these two gradients, they will know which part is anterior and which part is posterior. And when we have a mix of the two gradients, then we're going to have two different sections. So normally bicoid, the one that we find here in the anterior section, is a transcription factor. And it creates this gradient in concentration across the syncytium, and it's combined and diffused. So the mRNA degradation goes from the front to the back and is the least in the posterior part. The caudal does the same, but in the opposite direction. So bicoid and caudal are considered morphogens because they can occur in a concentration gradient. So every time that we talk about morphogens is because you have a concentration gradient, highest in one portion and less in another portion. And you're going to see this. Some of the molecules that we're going to be seeing through the course during the semester, we say they are morphogens, but they are also transcription factors. Okay? So just be careful with that. So at the end of the day, no embryo uses one type of cell specification only. Drosophila uses the three: autonomous, conditional, and syncytial. So, and as I say, same for the human embryos, some mammals, some vertebrates, they use at least more one of the other one. So in summary, we have the autonomous specification predominant in invertebrates, conditional in vertebrates and invertebrates, and syncytial mainly in insects. In the autonomous specification, the specification is acquired by the cytoplasm molecules present in the egg. So we're going to have transcription factors, we're going to have morphogens, okay? And cell type specification precedes any large-scale embryonic cell migration. So we need to have it at the very beginning to be autonomous, okay? Conditional, the specification by interaction among cells, so they require other cells and the position of those cells relative to one another, okay? If the distance is too far, the molecules that one produces won't affect necessarily the fate of the other one, okay? We're gonna need a huge amount of arrangements and migrations to complete that specification, but it allows us to have a regulative development, so they don't miss parts. So in the at the end, we have the cells differentiation allow cells to become a unique cell type. Cell goes under undifferentiated to a specific type of cells by committing to a fate. And cells can use those three different types of the specification. Autonomous, where the embryo process has the cytoplasmic determinants that function to commit towards a fate. Conditional is the acquisition of cell identity based on position, interaction with other neighboring cells, but it comes in contact with. And then finally, the syncytial, where the nuclei are maintained in a specific position, and the morphogens to which they are exposed finally determines the fate of those cells. And that is what I have for this chapter number 2. Okay, I need to go to try to open. There we are. Do you have any questions regarding this chapter 2? At any time, please feel free to ask any questions. I always say that if you don't understand right there, then you forget, and then there's way more questions just because we didn't ask right on there. Okay. Chapter 4 talks about cell-to-cell communication. So right now we ended up with how we can specify some cells, but then we need to talk also about how these cells communicate, right? So how we can determine the fate of these cells. So in the past decade, the development of developmental biologists, I should say, have started to answer some of the most important questions of natural sciences. We are beginning to understand how organs are formed. Okay, I need to go to my other screen, sorry. Go back. Go on, back one. Okay. So this is part of a bigger concept for morphogenesis, how parts of the body are formed. So a body is more than a collection of randomly distributed cell types, right? Development involves not only the differentiation of cells, but also their organization in a multicellular arrangement. And for instance, we have a finger and the hip section have bones, muscle, tendon, ligaments. However, their function and their structure is completely different. But it's the same type of bones, it's the same type of muscles, it's the same type of tendon, cartilage, etc. So when we examine the detailed anatomy of the organ, we see precise arrangements of multiple cell types, as I'm saying. So there should be a way they know, in quotation marks, where to go and what to form, right? Because it's the same elements, muscle, bones, etc. So they have a way to organize themselves in an orderly manner, take different shapes and make different connections. So this is what we know as morphogenesis, how they organize themselves, how they interact to one another to become functional. Okay, so how this organization can come about. So here we have, and in order to communicate properly among cells, they need a molecular communication. And this is where a lot of the students are going to say, oh my goodness, a lot of cell bio. And as I say, don't drop as of yet. This is the toolbox that I want to give you, okay? Don't be afraid. We're going to repeat this until the very last day of class. So today it may look overwhelming. Tomorrow also may look overwhelming. But we're going to repeat this again and again until I know it will sink, because I know it will. So This molecular communication is made through a highly diverse and specified protein-protein interactions, as we see in the figure. So remember that I say Juxtacrine signaling, okay? And for Juxtacrine signaling, the cells gotta be in direct contact, okay? So this is what we have here in A, the Juxtacrine signaling. Involves the communication across short distances and between neighboring cells alone. So cells have to be in contact, okay, as we see, and short distances, because we can kind of hold arms across the room, right? We need to be nearby in order to do it. So in this type of communication, the signal is carried out via membrane receptor, that this is what we see in here. Okay, these little wigglies here, that is the membrane receptors binding proteins in the and normally are associated with the extracellular matrix. Okay, so this gray section here. Okay, and they bind together. So we have here the homophobic binding, and here we have the heterophilic binding, and this is the extracellular matrix that allows this interaction. So they pass information to one another, and we're going to be seeing some examples of this today or tomorrow, maybe. Okay, all depends how much I cover today. In B, cells also communicate across distances. However, this can be a bigger distance. Okay. Here they need to be together. In here they can communicate across a little long distance through the secretion of proteins into the extracellular matrix. So here we have the signaling proteins that will act as a ligand. So this cell here is producing the signaling molecules that will be used by this other cell that is at a certain distance from one another. Okay, and this cell has a specific receptors for this specific molecule. Okay, so this type is called paracrine, and in this type of signaling, a cell secretes the protein and into the environment, and then another cell will be receiving that signal. So we will see later how the binding of the ligand to a receptor alters the shape, alters the conformation of the receptor, and this whole process has the ability to activate enzymatic reactions that constitute the signal transduction pathway. Okay, some of you have already had cell biology, right? When you talk about a lot of these signalings and pathways. So this is the sense. So the signal protein, the ligand, will bind specifically to a receptor. This receptor here gonna change gonna transform, gonna become active, and then there is a cascade of events that can generate change in this cell. Okay. So keep going. So there are 5 major questions from embryologists who study morphogenesis. The first one is how are tissues formed from populations of cells? So for example, the neural retina cells stick to one another, neural retina cells, rather than becoming integrated into the pigmented retina or the iris cells. So they just can bind to these neural retina cells only. The second question is, how are organs constructed from tissues? For example, and I use a lot of this example of the eye, is the retina of the eye formed at a precise distance behind the cornea and the lens. We don't flip it, otherwise the eye won't be functional, right? So the retina will be useless if it's formed behind a bone or in a kidney. So they have a specific positioning. The third question is, how do organs form in particular locations, and how do migrating cells reach their destinations? They need to know. There should be a highway directly to bring you to. It's not about going into the bush and then going back and forth and get lost. There is a pathway that they need to follow. Again, the eye develops only in the head. So how are cells instructed to travel along certain routes of the embryo, and how are they told to stop once they have reached their destination? It's not like the GPS that we have, right? Take a right, take a sharp left. You have arrived to your destination. So they need to know that they need to stop in that section. Number 4 is, how do organs and their cells grow, and how is their growth coordinated through development? Example, what controls the rate of mitosis in the intestine? If the intestine generates more cells than it should, it could produce tuberous outgrowths, right? And we know that the epithelium of the intestine is renewing constantly because there is a lot of abrasion, but that is very well coordinated, okay? And the last question is, how do organs achieve polarity? Polarity is a very important concept in development. So how is that the same type of cell can be arranged in different ways in different parts of the same structure? For instance, if you look at your finger, right? A cross-section of a finger shows the same tissues: bone, cartilage, muscle, fat, blood, neuron, and skin. But it's a cross-section of the forearms have exactly the same structures. But the tip of your finger also have a nail. You don't have nails anywhere else than at the tip of your fingers, right? So how they achieve that polarity, how they know which is the distal and which is the proximal part, where is the dorsal ventral, right and left. That is polarity in this case in embryology. So they need to know exactly that you will become ungual cells to become the nails. So all of these questions concerning cell behavior. There are 2 major types of cell arrangements in the embryo. We have talked about them. Epithelial cells that are tightly connected in layers, sheets, or tubes. And then the second one is the mesenchymal cells that are unconnected to one another and operate as independent units. So within these 2 types of cellular arrangements, epithelial and the mesenchymal cells, morphogenesis is brought about through a limited repertoire of variations in cellular processes. The direction and number of cell divisions, cell shape changes, cell movement, cell growth, cell death. Even though we don't like to think about the death of some cells, as I explained it on Monday, is important. We need cell death, programmed cell death, in order to open up some cavities and things like that. And finally, changes in composition of cell membrane or secreted products. So although the repertoire of morphogenic behaviors is small, only these 6 events, processes in here, cells can do a great deal with this limited set of instructions. So differential cell affinity. So many of the answers to the questions about morphogenesis involve properties of the cell surface. Even though we don't think too much about what the cell surface does, it does help a lot to coordinate the communication among the cells and what type of tissues they may form together. So the cell surface looks similar in all cell types. And many early investigators through that, the cell surface was, they thought that was not living. It didn't accomplish absolutely any role to the formation of the embryo. But we know now that each type of cell has different sets of proteins at the surface, and that some of these differences are responsible for forming the structures or tissues and organs during development. Okay? So what we see in this image are some experiments that were done by Townsend Hofstede back when in 55. So these two scientists that I have here at the bottom in 55, and they were using neural tube stage amphibian embryo. Okay, so we have an embryo that have already gone into neurula, and we know that it's neurula because we have the neural plate right here. Okay, and here, this is the neural plate. So this is what we're going to be forming the neural tube. Okay, so they took cells from the ventral part and cells from the neural plate. They dissociated those cells and then they mixed them together, okay, with dissociating and presumed neural plates and another part of that embryo. And then they mix it and they look at how they reassociate together, okay? So the mixed cells became especially segregated, okay, all around. And then they reaggregate. And the way how they reaggregate was that the epidermal cells were outside, as we see in here, while the mesodermal cells, that were the ones that were supposed to be here in the ventral part, okay, migrated centrally to be part of the neural cells. So the cells knew where to go. When having only cells from two germ layers, ectoderm and mesoderm, they knew where to place. Ectoderm needed to be exterior, and endoderm, mesoderm in this case, needed to be internal. But that was only in a very simple arrangement. In this other figure, we have a little bit more. We have now the three germ layers, ectoderm, mesoderm, and endoderm. And when they start doing a combination of cells, a group of cells that belong normally to each of the different parts of the cells and the embryo, they found that cells sorted out to their appropriate regions reflecting their proper embryonic position. So these experiments were made in amphibians, with amphibian cells, from the three germ layers. So mesoderm normally migrates centrally with respect to the epidermis, and epidermis we know is the ectoderm, and adhering to the inner epidermal cells, and that's what we see here in A. Epidermis and mesoderm, we know that the epidermis is mainly ectoderm, so they aggregated them and they resorted, and then ectodermal cells become outside, becoming the epidermis, and the mesoderm went inside. Okay? Mesoderm, endoderm, they rearranged the same again, but in this case, what happened? The endoderm should have been inside, right? And it didn't. And it's just because it's missing the signal from the ectoderm. Okay? So we have a different ways how they arrange, but they know more or less where they need to go. In this case, we have the three, epidermis, mesoderm, and endoderm, that rearranged, and ectoderm became outside, mesoderm in the middle, and endoderm in the inside. So these findings were interpreted in terms of selective affinity. That's the term. Okay, so the inner surface of the ectoderm has positive affinity for mesodermal cells and a negative affinity for endoderm cells. So selective affinities change during development. For development to occur, cells must interact differently with other cell populations at specific times. It all depends when organs are created or tissues are created. And this is what we see here. So from the previous slide, we know that cells do not sort randomly, right? They're going to aggregate based on their affinity with the other cells surfaces. And so instead of they move to create a different tissues and organs, but now they solicit the inclusion of new terms, and is the differential addition hypothesis. And this is what we see in this image. So certain cells migrate centrally when combined with some cell types, but migrate peripherally when combined with others. And that's what happened in this case when I say mesoderm and endoderm, because endoderm should have been inside and mesoderm should have been outside, right? In the position of an embryo. But because we didn't have the ectoderm, the mesoderm got confused. Okay, so cells have the tendency to aggregate, with the smallest interfacial free energy. Okay, this means that cells are rearrange themselves not in the into the most addition. So here we have A and B, and here we have B and C, and here we have A and C. So different tissues. A has a greater affinity than B. In this one, B has a greater affinity than C, and A greater than C. Okay. So depending on this type of rearrangement, here we have the surface tension, and the way how they arrange is with the less energy. It doesn't cost that much to have the affinity. Okay. So now all that is required is sorting to occur, is that the cell types differ in the strength of their addition, which means that a sort of glue to put them together. But at the same time, this glue seems to vary among the cell types, so not always going to be rearranged in the same manner. It all depends. They always need that interaction, and that is why the connection, that affinity of the different cells with one another allows for the formation of different tissues. And I'm going to leave now here, okay, because we're going to start talking about the glues. What allows one cell to glue to one another and to form tissues. So I'm just going to stop sharing here. Okay, so once again, I will be teaching tomorrow during the lab session. So if you are at 10, please be at 10. If you are at 1, so I will see you at 1. So far, I have 3 students at 10 a.m. I think I have sent emails to see if those can be changed to 1, because my TAs can only be at 1, otherwise it will be me that will be with you. So if you are at 10 a.m. and you can change to 1, that will help me a lot. If not, I will be there, okay? Because I know that you plan your schedule based on those 2. Initially, we didn't know whether we will be in person this semester, and that's what the setup of the labs were made that way. But since we are virtual, if you can change to 1, that will help me. If not, I will be there with you. I will be happy, okay? I'd rather have a small lab so we can have better chats and I get to know you better. When you are in a big crowd, I don't even get to know your names normally. I am missing that so much, because at the end of the semester, I know your faces and your names, and I can associate it. But here, if you have you don't even put your camera, I don't know who you are, right? So in the lab, I prefer to have small groups too. But because we are virtual, we need to suck it up. And it is what it is, and we're going to do our best. So if you are in the 10 a.m. and you can change, please do so. I have not received all of the groupings. So remember, for the first assignment for the video, if you have a partner, please send me an email. Otherwise, by tomorrow, I will be pairing you. Okay? So you have until today at 5 p.m. to let me know with whom you will be doing your video. Otherwise, by tomorrow morning, I want to have a list of the ones that I don't have pairings still, and I will pair you. Annalie, can you send me again send me again the email, because it's buried among the 55 emails that I got yesterday. So send me that email again. There are some people that haven't registered still to perusal.com, so please do so, because we're going to start using that one for looking at the interaction among the different groups, okay, for their readings, for the participation in the discussion, okay? Any questions? No? Everything is clear so far? Good. So I wish you have a very good afternoon, and I will see you tomorrow in the lab session, okay, for lecture. Yeah. Bye-bye.
Thank you.
Thank you.